This video is going to be all about the inside of a prokaryotic cell. So the space inside of the cell is known as the cytoplasm, and it is actually filled with a jelly-like fluid known as the cytosol. And it's that cytosol that kind of keeps everything inside the cell afloat. Okay, so it prevents everything from just dropping down to the bottom of the cell. So the inside of this cell is mainly water, so about 80% water. It's also got various proteins, carbohydrates, lipids, ions, all the things that a living thing needs in order to live. So suspended in that jelly-like fluid, the cytosol, is going to be the DNA, right? The genetic material of that bacteria. We also refer to that collectively as the chromosome, but more on that later. So we're going to have the DNA, again, genetic information, and then other structures known as ribosomes, okay? And occasionally, bonus feature that some bacteria have are plasmids. So let's talk about all three of those things a little more detail on the next slide. So all living things have DNA as their genetic material, and all the DNA together is known as the chromosome. So the bacterial chromosome is a circular thread of double-stranded DNA. And the reason DNA is called the genetic material is because it includes the directions that the cell needs to make proteins, because proteins are the workers of the cell. So because the DNA is inside of the cell, in a region that we refer to as the nucleoid, because remember, prokaryotes don't have any internal structure or internal organization. All the stuff inside the cell is just kind of hanging out in there together, chilling in the cytosol. So where the DNA tends to hang out, kind of all condensed together, is called the nucleoid. And also in that jelly-like fluid are going to be ribosomes. Ribosomes are the protein-making machines. So it makes sense that they're in there with the DNA, which are the directions to make proteins. Okay? So ribosomes actually come disassembled. They're made up of a small subunit and a large subunit. And when it's time for the cell to make proteins, they assemble together to form a complete ribosome. And you don't have to worry about the numbers 30S, 50S, 70S. Later, we will be talking about how the ribosomes in prokaryotes are a little bit different structurally than the ribosomes in eukaryotes. They have the same goal to make proteins, but they do look just a little bit different from each other. And that will be important later when we talk about antibiotics and what they target and how they work. So inside the cell, chromosome, right, all the cell's DNA, and again, it's circular double-stranded DNA, or I should say double-stranded DNA that kind of forms a circle. The ribosomes are protein-making machines, and some bacteria also have plasmids, okay? So plasmids are described as extrachromosomal, because that word extra means it's separate from. Okay, so plasmids are pieces of DNA. They are also circular, also double-stranded DNA, but they are not part of the chromosome. Okay, and because they're not part of the chromosome, that means they're not essential for life. So they do not include any crucial or essential genes. Rather, they're bonus features. They offer the cells some sort of advantage. So for instance, some bacteria that have a plasmid are also resistant to certain antibiotics, because on that plasmid are the directions for making some sort of protein, perhaps, that can break down certain antibiotics. Okay, so it's just an advantage that the plasmid offers to the cell. So I do kind of want to reiterate, all cells must have a chromosome. They have to have DNA, and they have to have ribosomes, because without DNA, you don't have proteins. Without proteins, the work of the cell doesn't get done and the organism would die. Okay, so chromosomes and ribosomes are absolutely required for any living thing. Plasmids are extra. Okay, not all bacteria will have plasmids, but some might. Okay, so it's just a bonus feature. All right, y'all, that is it for this video, short and sweet. Let me know if you have any questions.